Now let us see the acquired drug resistance. So under this actual modification, we are going to look at the acquired one now. Acquired modification. Now here what happens, there is development of resistance by an organism. So the organism develops the resistance. So it was sensitive before and it is not sensitive now. Okay, first of all, I think we need to fill all those details for you here. Wait. Go back here. Acquired means what? Earlier it was microorganism was sensitive. Okay, but now it is not. Earlier microorganism was sensitive, but now it is not to a particular antimicrobial agent. This is what is called as the acquired drug resistance. This is the problem. This is a issue. This is a real problem. Now, where exactly have we seen these type of resistances? Example, Staphylococcus. You remember in Staphylococcus you have studied MRSA, vancomycin resistant Staphylococcus aureus. Finally, you will give linezolid, correct? Similarly, in tuberculous bacteria, Mycobacterium tuberculosis, also you have seen drug resistance, correct? These are very rapidly acquiring drug resistance. Also coliforms, they acquire quick drug resistance. There are some other bacteria which don't develop significant resistance. Those are going to be, example, Streptococcus pyogenes and spirochetes. These don't develop much resistance. This is a green for us, right? Very good. Now let us see how exactly the drug resistance is achieved. It can be either through mutation or gene transfer. Mutation can be a single step mutation or a multi-step mutation, and a gene transfer can happen via conjugation, transduction or transformation. Now do you know what mutation is? The DNA of the microorganism or the genetic material of the microorganism itself can undergo mutation. Okay, so it is not acquiring any genes from outside. However, in gene transfer, the genes are coming from outside. One bacteria which is resistant to a particular antibiotic goes and tells another bacteria, hey, look, I have a copy of a gene which is going to help you become resistant to that antibiotic. Will you buy this from me? And that bacteria is like, yes. It's like an antivirus software that you buy for your laptop. These bacteria land up making each other resistant to the antibiotic. So this is like conjugation, what you see here. So in conjugation, what is happening? There is a pili, okay, a sex pilus or a bridge is there, okay. These are some keywords. It is a pilus between two bacteria. In transduction, you land up actually having bacteriophages or virus which are going to transfer the gene into the bacteria. So you can call that as a bacteriophage or a bacteriophage, however you want to call it, you call it. Then transformation. Nice background music, I understand. Please bear with me. Transformation means, see, one bacteria will release the gene into the medium and the other bacteria will pick up the gene from the surrounding medium. Pretty simple, right? I don't think it's so complicated. 3 ways of gene transfer: conjugation, transduction, transformation. Okay, moving on. Drug resistance, there are a lot of things. It can get tolerant to the drug, it can destroy the drug, it can be impermeable to the drug. So these are what action it actually the microorganism can have on a drug. Details are in KD Tripathi. Okay, we will go through the details if we get time in another video. This video is already overflowing. Then there is something called as cross resistance. Let me just tell you what cross resistance is. This cross resistance, no, it is actually a bigger problem. The organism is becoming resistant to a drug to which it was never exposed. The organism has not been exposed to a drug, but it has become resistant because there is something called as cross resistance. If it was exposed to antimicrobial agent A, it will become resistant to antimicrobial agent B. Like example, if you have, like, what are tetracyclines that you know? oxytetracycline and doxycycline, right? The thing is, if you expose it to doxycycline and it becomes resistant, it automatically becomes resistant to oxytetracycline also. Now let us say sulfonamides. So if one sulfonamide it is resistant to, it means it will get resistant to all other sulfonamides. The sulfonamides we have studied are sulfadiazine, sulfones like dapsone, para-aminosalicylic acid, that is PAS. Okay. So if it is resistant to one, it gets resistant to all others. Now currently where are we? Currently we are here. See, whatever example we have given you, these are complete. That means one, if it is resistant to all others in that group, it will become resistant. This is complete. But there is something called as partial. Lot of examples in textbook. I request you to kindly look at the textbook for more information. Now only two more topics left that we want to cover. Drug resistance, how you will test, and drug resistance, how you will prevent. Testing, you can do a lot of methods. I'm just putting one example here. Kirby-Bauer disc diffusion method. Here basically you will put a lot of antibiotics on one plate and see whether the bacteria can grow. Like in this case it can't grow, but here it has grown. So here it is resistant to bacitracin. However, it is susceptible to tetracycline because it's not able to grow around tetracycline. Okay. Is it clear? So let me repeat just in case. If it is not growing in near the antibiotic, it is susceptible. The antibiotic is able to stop the growth of the bacteria. Here the bacteria has grown around bacitracin to show that it is resistant to this antibiotic. That is, bacteria is overpowering the antibiotic. Here bacteria is not able to overpower the antibiotic. Okay. Now, how will you prevent drug resistance? You should not have any indiscriminate use. You should not prolong the use. Then inadequate also you should not give. So it's like a, you should correctly balance it and give. Then you should make sure that you give rapid acting antibiotic, narrow spectrum preferably, don't give broad spectrums. Then use combinations. Combinations you should use for prolonged therapy. You should use combinations like for AIDS, tuberculosis, you should give combination drugs. And then if there are organisms which are notorious for developing resistance like Staphylococcus aureus and tuberculosis, then you should give intensive treatment. Please just look at all the slides that you have read so far. This one is important, how it will become resistant, actually what it will do to the drug. Cross resistance, testing and prevention. Bye bye.